All right, well, thanks everyone for attending today. I have the pleasure to tell you about BioAge. We're a clinical stage drug discovery company and drug development company where we target fundamental mechanisms of aging to treat diseases of aging. And I'm going to talk to you about biomarkers. We are very passionate about our human-first discovery platform, where we use over 45 years of longitudinal human molecular and phenotypic data to identify translational targets in humans, so targets where we believe drugs against those targets will work on aging in humans. And I'm going to tell you the story of how out of that platform arose our current lead program, which is a muscle aging drug, BGE-105, where we have some very exciting phase 1b data on how it prevents muscle atrophy. And then how we are going to use that data to pursue preventing muscle loss in obesity. When people are trying to lose weight in obesity, they lose muscle, and we would like to prevent that. We also have an exciting immune aging program at the bottom there, but I won't be talking about that today. So we know that there is this tremendous variation in the human lifespan, not nearly as much as across the entire taxonomy of mammalian species, as Steve was talking about, but nonetheless, there are people who live incredibly long and people who live incredibly short. And these are the natural examples around us of essentially more and less robust physiology, fast and slow agers, if you will. And this is the framework that we use to look at human molecular data to identify pathways that can make more of us live like the very robust, healthy agers who can live in a very good condition to a very old age. This is, Robert Marchand, who was the namesake for BGE-105, our muscle aging drug, who was competing in cycling at age 105. Be awesome if more of us could do that. So we're a very data-driven company. We begin looking in a hypothesis-free way, looking at very large-scale omics data across thousands of patients, and we generate millions of data points from these patients, model them longitudinally over decades to look at lifespan and key health span outcomes to figure out what are the targets we want to modulate to really do something impactful for human aging. These are the kinds of things we model. So all-cause mortality, cause-specific mortality, and then these key health span phenotypes on the right: cognitive performance with age, incidence of diseases, muscle function with age. So all the things that you'd want to look at if you wanted to say that someone was aging robustly in a very healthy way and were functional. We can look at this data in a lot of different ways. Here's one very simple way of visualizing 5,000 proteins. Each of these circles is a protein. And they're plotted here with respect to their correlation to longevity over 30 years of follow-up. But that's the y-axis there, hazard ratio. But you can see there's a diversity of associations with respect to these health span phenotypes. And so these circles are colored differently depending on whether there are robust associations with each of these 4 phenotypes on the left, things like physical function with age, brain aging, renal function, cardiovascular. You can cut this many different ways, but it begins to give you a sense of the complexity of the modeling and the way we can model different phenotypes with aging. Another emergent property of having so many different measurements longitudinally over time is we can begin to model the relationships between molecules. And so this is what you see here. When we look at how all of these proteins change from person to person and across time, we begin to derive data-derived pathway analysis, essentially. So you can see a cluster of unordered data self-organizing in a sense as we do clustering analysis into related clusters, which we can then prune to remove spurious correlations, and then ultimately subdivide into sort of mechanistically unified, in some sense, groups, which we can begin to characterize. And this helped us map the landscape of aging biology. And that leads to a very interesting conception of how we view this platform, which is based on high quality, rigorous, longitudinal human data, which we are using in a hypothesis-free way to look at aging mechanisms. And one way of looking at this is that clearly there's a bunch of really interesting aging mechanisms which have been elucidated over decades, which I'm representing here in the hallmarks of aging circle. And a lot of that work was done in non-human species. And so this is a lens to say, well, which of those mechanisms are actually conserved in humans and we believe will be translational to humans? And then hiding behind that circle on the right are all the things which maybe haven't been seen yet in model organisms, but are emergent, either because we haven't studied them yet or because they're unique to humans. And so there's a number of exciting ways to use this data. And to be clear, my talk is about biomarkers. So here you've seen how we're measuring all these aspects of human biology, these biomarkers. And our aim is to prioritize what initially looked like biomarkers into the most likely causal pathways so we can modulate those with drugs. And then I'll tell you the story of how we then measure aspects of the clinical trials where we put those drugs into people to apply those biomarkers back to the platform. So we have a focus right now on muscle and metabolic aging. We're also interested in immune and brain aging. And I'm going to tell you the story of our lead muscle aging drug, which is based on the Apelin pathway. So this is, there are a lot of different analyses we do to look for targets, but when we were looking for a muscle aging target, one of the things we did was construct a muscle aging score out of a lot of these phenotypes I've been talking about. And when we drew the distribution of that, this is on the upper left here is the distribution of scores that came out. And you'll see Apelin residing on the right-hand tail of that distribution. So one of the strongest hits. And at the time, we were interested in licensing a drug, so taking a drug that existed and using it to combat muscle aging. And this was essentially the one reason it was interesting. It was the most robust signal that we saw that also had a drug that we could potentially get our hands on. And when we looked deeper into that relationship, we discovered what you can see on the right here, which is if you just look at Apelin levels on the x-axis, you can see that they correlate in a robust and essentially linear, like continually increasing way with better probabilities of good outcomes with aging. So longevity there is modeled as the ability to, or the likelihood of living to over 80, and preservation of grip strength is living to over 80 with robust grip strength. So this has a really encouraging relationship with these two key outcomes with aging. And that made us wanted to look in a lot more depth. One thing we did is model the Apelin signaling pathway in our data-driven way. And we can see it has a really nice association with all-cause mortality on the right, where the higher the levels of Apelin pathway activation, the lower your mortality. After getting so enthused about all of this human data, we went and we talked to Amgen, who had this drug we were interested in that modulates the Apelin pathway. And we asked if we could have some of it to test in our models, and this is the result. So we first put this into mice over 60 days. They were very old mice, 20-month-old mice are quite frail. And you can see their distance run per day declines precipitously just over 2 months at that age. But if we put BG105, which activates the Apelin pathway, into their drinking water, those curves begin to diverge, and they do so in a very statistically robust way. So that was highly encouraging. You can see other data on the right here from that study where if you look at grip strength change, grid hang time change in these mice from the beginning to the end, you can see the change plotted here. And mice on vehicle are all below the line, saying they all lost. They had a decrease in grid hang time. They couldn't hang as long, so they had less strength. Whereas the BG105 treated mice were basically preserved over that entire time. So this was really encouraging. But the next study is what actually helped us figure out which way to go with this drug, where we took mice and we casted one of their hind limbs, and when the limb is casted, it doesn't move, and the muscle shrinks over time. It's atrophy. We all get this. If I put myself in a cast, it would be the same. And we saw this really interesting result on the right, where if you put these these legs in a cast, they really atrophy quite a bit. But if you give them drug, they atrophy almost not at all. And you can see one leg was casted and one leg wasn't, so they serve as their own internal controls. So this was a really powerful result that made us think about what we could do in humans. First, just to review a little bit of the pathway and the drug. So Apelin is known now as an exerkine. So it is released in response to exercise and has signaling functions across the body, particularly muscle, heart, brain, and has some mechanisms which are being elucidated, which include increased regeneration, stem cell activation, decreased atrophy, as I just showed you, and decreased inflammation. And we in-licensed this drug from Amgen, which targets the apelin receptor. It is an oral drug or an IV drug, and has good safety data, very well tolerated drug. And so we decided to put this drug as our first clinical evaluation into a phase 1b study in healthy elderly volunteers, so they don't have any disease at baseline, but they, like all older people, are at risk for muscle atrophy. So if an older person gets admitted to hospital for any cause, really, they are at tremendous risk for atrophy. And the older you get, the quicker you atrophy and the harder it is to recover from that. And that's very robust across a wide variety of clinical circumstances. But if you just take healthy older people and you ask for volunteers, they volunteer to be at bed rest for 10 days. So they're going to lie in bed all day and only get up to go to the bathroom. They experience substantial muscle atrophy, especially in their limbs, their lower limbs, legs. And what we found is that, much as in the mouse study, which was really exciting, that BG105 robustly prevented that atrophy. So whether you look at thigh circumference, just with a tape measure, or individual muscle sizes in the thigh or calf by ultrasound, you found very significant protection against that atrophy, greater than 50 to 70% protection against the atrophy that they would have otherwise experienced during this period. If you look at the muscle quality with ultrasound, that's the third one there, at bed rest, people's muscle quality declines. Their muscle becomes more heterogeneous and infiltrated with fat, and we almost entirely prevented that change in these people. And then we also got a peek at the mechanism, which I'll show you in a bit more detail in a second, which is that we also robustly preserved muscle protein synthetic rates. So when you go at bed rest, it's actually a really robust, it's a powerful physiologic stimulus being at bed rest. It changes many, many things about your physiology. And one thing it does is cause a precipitous drop in muscle protein synthetic rate. And that's tightly linked to the fact that you lose a lot of muscle. You're not synthesizing new muscle to replace turnover. And we preserved that muscle synthetic rate. So here is the muscle synthetic rate data. I'm focusing on a number of the molecular measures that we found here, and we'll see how we follow up on those. So you can see across a wide variety of proteins in the middle there. In almost every case, we have differences, most of them statistically significant, where people on drug had higher synthetic rates for each of those individual proteins than people off drug. And on the right, if you do a normalized version across all of those proteins, we have this highly statistically significant result. This is pretty awesome. So as I described, we have this longitudinal human aging data where we discovered this target. And we have these various signatures of disease, signatures for these things on the left, like strenuous activity, over time, longevity, whether people live a long time, whether they have good walking speed, over decades in the future. So we have all these different signatures. And if you look at the proteomic changes in people on drug in this trial versus off of drug, it shifted their proteome in a way that is beneficial for every single one of these protein signatures on the left. So the drug that came out of our platform modifies the proteome in a way that is making people physiologically healthier, younger, according to these biomarkers. So not only are we inducing this very interesting acute prevention of muscle atrophy, we're shifting all of these biomarkers in a way that says that if they were to have this physiologic state for decades, it would be really good for all these things on the left. On the right is just a detail of just one of those categories, so you can see some actual data. So for walking speed, so each of these changes was highly statistically significant that was induced by the drug, which are those p-values there. But the way we do this is for walking speed, for example, we have all these individual proteins. They either have a significant positive or negative correlation, meaning that higher levels of those proteins was good or lower levels of those proteins was good. And then we plot out the changes on the bottom there. You can see how those curves diverge and how the drug caused a net positive shift in the good factors and a net decrease in the bad factors. So pretty interesting data. So that's one way you can use biomarkers that we derived. You can also use external biomarkers or biomarkers validated on specific gold standard clinical outcomes. And so these are soma signal tests. So SomaLogic is the company we use to do our proteomics. They're leaders in this field. And one of the things they do is they use large cohorts to validate proteomic surrogates of gold standard outcomes, in this case, resting energy expenditure and cardiorespiratory fitness, or VO2 max. And so they've derived predictors and external cohorts and validated those predictors, and now we're just applying that to our proteomic profiles. So using their models, we're seeing these really interesting trajectories where, again, you see this really powerful effect of bed rest, that people on bed rest drop precipitously in their resting energy expenditure. And that correlates directly with loss of muscle and lower metabolic rates. And that's almost entirely preserved on drug. And the same thing with cardiorespiratory fitness. Drops precipitously on bed rest and is preserved on drug. So it's beginning to create a very consistent package of data, as you might notice. We also, the muscle synthetic rate data that I showed you was based on muscle biopsy that we did at 3 time points during the study. And on those same biopsies, we also did single nucleus RNA-seq. And we found a number of things. We found data in the muscle cells that recapitulates what I showed you in the synthetic rate. So rates of rates of synthesis of transcript are also preserved on drug. We also found very interesting data related to lipid droplet synthesis in fat cells, which relates to that obesity indication I mentioned as well. But what I'm showing you here is the tremendous effect on mitochondrial physiology. So you can see, I'm going to talk about PGC1 alpha, often known as the master regulator of mitochondrial biogenesis, where we again see if you're on bed rest, your mitochondrial function drops a lot, and if you're on drug, it's preserved. And we saw this consistently across all the respiratory complexes that you see here. So all of these biomarkers in humans helped us find a target that we then tested in mice and brought into humans and tested another bunch of biomarkers, which are teaching us what to do with this drug and helping us in a number of ways. So basically, these phase 1b results have unlocked a range of potential muscle-related indications that we could pursue clinically. Our lead program is going to be in obesity, where as you know, there's a tremendous popularity of GLP-1 drugs, which are very effective at inducing weight loss. Interestingly, a large percentage of that weight loss is muscle. Somewhere between 30 and 50% is lean mass rather than fat, as you might expect. And the older you get, guess what? The larger fraction of that weight loss is muscle. And then, of course, the harder it is to rebuild that muscle. So this is a big risk. The older you get taking these drugs, the more likely you are. So first you start out with a lower muscle mass, the older you get, and then a larger fraction of your weight loss is muscle. So there's a strong risk of inducing frailty. While of course you're doing these other beneficial things at the same time. You're improving your metabolic health, but you're decreasing your muscle health. So it's a very compelling case for trying to prevent that. We're also planning an ICU study where we're trying to prevent diaphragm atrophy. So the diaphragm keeps you alive by breathing. If you're intubated, it doesn't work, and it atrophies. And that can prevent you from being able to leave the ICU alive. Same thing with peripheral muscle, especially in COVID. There were a number of very prominent examples of people going in with very robust muscle function and then coming out with, really wasted. And that happens to everyone who goes into the ICU for any length of time. So we hope to be able to prevent those. So here's the story of biomarkers from our clinical trials. So we took longitudinal human biomarkers and used them to lead us to where we are today. And the biomarkers from these trials really help us in all these ways on the right here. So really to elucidate the effects on human biology, kind of confirm existing hypotheses, also generate new hypotheses that we wouldn't have expected. I mentioned a couple of those. We can look directly back at aging pathways, based on our own research. Like I mentioned, all those age-related physiologic deteriorations, which we can now map back to. And we can also look for new clinical biomarkers, so ways to select patients who might respond better or measure their treatment response, and I showed you some of those as well. So biomarkers are really a compelling way to go about really finding translational targets and improving clinical development approach. That's it. I'd love it if we could all help patients die young as late as possible.
Thank you, Eric. Questions?
Eric, that was really encouraging. I'm really happy to see how well the thing's going. So overwhelmingly, the benefits that you're finding from your drug are preservation of muscle mass and strength and function and all that, as opposed to restoration. So I'm wondering whether you've thought about or perhaps even done some experiments to look at co-administering your drug with other interventions that restore muscle mass, but perhaps, don't restore muscle quality, like, growth hormone, for example. Mm-hmm. Have you done anything like that?
We haven't. We pursued a fairly focused development path. Interestingly, we don't know that this will not regenerate muscle. We just haven't investigated that very deeply. We found a very robust prevention of atrophy and realized there's this very direct path to development in prevention of atrophy. And it gets at also what you mentioned is that there's existing drugs which have been able to increase muscle mass but not actually function, or in some cases even strength, which is, of course, initially shocking, right? But here we really, in all the ways we've studied it so far, it appears that we're preserving not only the mass but also the function. Looking more into the sort of rebuilding of lost muscle is something that we plan to do in the future.
Hi. Is it a dose-dependent drug, like based on body weight or?
Absolutely, yeah. Mm-hmm.
Got time for one more question?
This is a quick one. Is there implication for neurodegenerative diseases, so Parkinson's, Alzheimer's?
There's a lot of potentially direct and indirect relationships to that. So I won't talk about everything, but just an obvious one is that the better your muscle health, the better your cardiovascular health. You saw the VO2 max, a lot of that's heart. And the better your cardiovascular health, the better your brain health. So I think there's a lot of really interesting possibilities there, but we're exploring or focusing on at least, the most direct benefits first. I think there's a lot of exciting ways to develop this long term. Mm-hmm.
Thank you, Eric.
All right, thanks.